We are the have great host and moderator of this event. And we welcome you all who are going to protect this world of who denies on the second day of National DNA Night National Team. We take the opportunity to welcome and introduce Dr. Day1 of you.
Dr. A. Mohammad is a faculty from the Department of Biotechnology and School of Biotechnology and Technology. He has done a PhD in biotechnology and a master's in science in bioinformatics from Day1 University. A major area of research includes structural biology, and publication has left a major impact on structural biology. Now, we would like to welcome Matt and
Thank you for your introduction. Good morning, all. So you had enough introduction about me. I am a faculty of VIT and it's not much required. Anyways, first of all, before I start my session, I thank Professor Radha Saraswati and her team, young and dynamic team, for having conducting like this kind of event for the 9th year in a row. This is not just a small event, right? So kudos to our entire team. And I also thank Professor Arnold and ma'am for inviting me to give a talk in this particular DNA day. So thank you all. Let us start the session. And yeah, this is the talk for today, protein-protein interaction network. So this is what a topic, and you might wonder why ma'am is talking about protein interaction for the DNA Day conference, isn't it? So we all know actually the DNA, I mean the ending point or the proteomics starts from where the DNA ends. Like it all goes hand in hand. DNA or the genome and the protein, they work coherently, isn't it? So that is what I have chosen this, and let's see how informative it will be. Okay. So why protein interactions? And we all know that our genome, we have our entire genome being sequenced in the year 2004. We have the Human Genome Project we proudly published. And that, I mean, the entire genome content or the entire DNA content of a cell is what we call as genome. Okay, so now that we have identified back and forth top to bottom of genome, and slowly the focus moved on to looking for proteins in our cell, right? So, and that also we have done with proteome, like the human proteome draft has been published in the year 2014 by Akhilesh Pandey and Bernard Custer. Like it was American and a German company, both together have published the proteome. So having in hand both genome and proteome, our focus is now turning towards interactome. You might be wondering what is interactome, because for many of us, this term is new, isn't it? So interactome, like from these two old terms, you would be very familiar that the word ome, it goes for the entire collection, the holistic content. So interactome means the entire interactions, especially the protein-protein interactions that is happening in a cell or in an organism, we term it as interactome. Okay, so now the focus is towards the interactome because these two components are much studied nowadays. Okay, so interactome is what we are going to see. More we will see about that in the coming slides. Okay, so this quickly we will see genome. The total amount of genome we have, like 99%, we have covered the data. Okay, and when coming to mRNA or transcriptomics, we have 80 to 90%. That is also near to 100. We have done it. We have our data, and it is accurate too. But now coming to function or especially interaction, we just have 30% of protein to protein interaction data that is known. And among that 30% data, 70% is spurious, like it may have errors in it. Okay, so we are very much devoid of this interaction or protein function data. So directly, you can relate protein to protein interaction to identify the protein's function. Okay, generally speaking, we are very much devoid of protein function only. Nearly 40 to 50% of the protein is not known for its function. We know that the protein exists, but we do not know why for is it expressing, what is its role inside the cell, what is its biological role, we do not know. Okay, so we are slightly, instead of studying that protein in a very greater detail, we are moving, we are coming outside the protein and seeing to what other protein does it interacts with. Okay, so having known the interaction, we may get a clue on its function, isn't it? So I'll tell you one good example. You will understand protein-protein interaction well. Say we have a protein A. That protein's A structure is known, function is known, like it is fully annotated. Okay, and now you have a new protein. You came across identifying a new protein in your lab, and let us name that protein as protein X. So you do not know what is its function. You do not know from which source it is arrived. Nothing you know. But incidentally, you got to know protein X is interacting with protein A. So will you now get a clue on its biological role and which pathway would it be involved in? Protein A is a well-studied protein, fully annotated, top to bottom information is known. Protein X is now interacting with protein A. Would you get any information about this orphan protein? Yes, because we all know protein to protein interaction or any molecular interaction that is happening inside our body is meant for any biological role. Okay, so without any meaning, this X would not come and interact with protein A. So if it is interacting, we get to know both the proteins A and X would be involved in a same pathway, isn't it? So that's how we identify the protein's function, by studying their interaction patterns. Okay, and that's how the work goes. Okay, the next slide. Yeah. So the availability of fully sequenced genome led to the large-scale studies of protein-protein interactions. So from here onwards, in our slides, we will be coming across this abbreviation PPI. So interactions between proteins are important for majority of biological functions. Not only just in terms or in context of function, none of a protein can do its role independently, isn't it? Do you all agree this? It cannot perform an entire biological role as a single entity. It will go interact with other protein. It will release some signal. That signal will go in, activate a next group of protein. Like that, it goes hand in hand. Okay? So like the association could be as small as just 2 proteins, or it can go up to 300 members interacting together to perform one biological role. Okay, as I was telling in the previous slide, the complete protein-to-protein interaction map is called as Interactome, and we have a specific database for it called as Intact. So this is the database for Interactome data. You will, like when you enter a specific organism name, or when you enter a specific tissue name, lung tissue or liver tissue, you will get the Interactome of, that particular tissues or organism. Okay, so proteins interact with other proteins to perform specific biological function. So here you can see 2 proteins are interacting, and of course, this is a host, like it can be a human protein, and this is a viral protein. I mean, this blue color is human protein, this is viral protein. So in order to establish an infection, the protein of a pathogen should bind to the protein of a host, isn't it? Of course, to make that also happen, there happens an interaction. And you see this yellow color area, this area is called as interface. Okay, interface is a region where the proteins, 2 different proteins interact. Okay, the residues that is required for interaction could be termed broadly as interface. And of course, we can call it in other terms like active sites or binding sites, etc. So some points about this protein-protein interaction, they are specific. Okay, none of a protein will go interact with other protein unwantedly. If at all there is an interaction, it is meant for a purpose, right? All right. So it occurs between a defined binding regions of a protein. That is what we call as interface regions. Okay? So interaction won't happen at any other sites of the protein. If it is happening, it will happen at this particular interface region only. All right. So, and of course, these interactions will have a particular biological meaning, right? Okay. So how do we identify the protein is interacting with other proteins or not? There are some experimental methods, wet lab methods. There are also equally computational methods to identify this protein-protein interaction. Okay, so a very classical method to identify protein-to-protein interaction is Y2H. This is a very old method, like it dates back up to 40 years, but still this is considered as an accurate method to identify protein-to-protein interaction. Y2H, have you come across this term? What does that stands for? Is2 hybrid assay, right? Okay, so this is considered as the best method, a classical method to identify protein-to-protein interaction. Apart from that, we have something called as bait and the reverse bait method. Okay, so we will quickly see what is bait, reverse bait. And have you all had this assay? Is this assay known to you, Y2H? Because this is coming in the slide. If it is known, I can just skip it out. Okay, let's see. Okay, so this is bait and the reverse bait method. So you can relate to a small example which I just gave while starting the class. I gave you an example. Protein A interacts with protein X, isn't it? So let's have this protein 1 is a well-known protein, known protein in the sense its structure is known, its function is known, well-known. So we are going to identify what are the other proteins that binds to protein 1, right? So what we will have to do, we will have to extract the protein, extract the components of a cell, enrich the proteins alone. You have to purify the proteins alone and incubate along with. And another important thing is this protein 1 is immobilized. Can you see some solid support? It is immobilized. It is stuck to some medium, right? Now you add a cell extract. So cell extract will have a putative protein partners. The partners of 1. So this 2, 3, 4 protein interacts with this particular 1 protein. Okay, so if that is happening, what immediately would you do? What is your spontaneous assumption? The protein number 2, 3, 4, which is unknown for its function, which we do not know why it is expressing, are now interacting with the known protein. So you get a clue that protein 2, 3, 4 are interacting one. So they have a similar function. Not considered as the end of the experiment because we need to confirm. Any wet lab experiment has to be repeated. You need to do replicas, isn't it? So we will have to confirm, like we first identify what is 2, 3, 4 using mass spec. That is all done. So then onwards, when you move on to the second step, we call it as reverse bait experiment. The first experiment is called as bait. This is called as reverse bait. So in reverse bait, we pick out one protein among the newest member. For example, 2 is immobilized, and then we add the same cell extract and we get to know if the same members are associating. All right. So you see again 1, 2, 3, 4 is there, and you come across a new protein 5. So it means your experiment is still not confirmed. You have come across a new protein called as, protein 5. So all of these may be performing the same biological rule, right? So now what will you do? Is the experiment done? No, because you have come across a new protein. So you will have to again confirm and immobilize protein 5 in the next reverse bait experiment. So if this protein is immobilized, you come across a different protein, like as many times as you come across a different association, this has to be repeated. Okay. And until when this experiment should be done? When you do not get any new members, if you repeatedly get same members associated with each other, it means your experiment is done. Okay, so this is called as reverse bait experiment. This is considered as a high throughput because in one experiment you will be able to pick out all the interacting partners, right? So this is how protein-protein interactions are identified. It is stored in the database. All right. Okay. So the next one is, the net result of this experiment is at the end we will be able to map several protein-to-protein interaction. So when you get a protein interaction, you will get clues on the functions. Okay, because we are very much devoid of function information. Say it a gene or a protein, whatever it is, we are lagging, very much lagging. There is a serious gap in identifying a protein's function. Okay? So this kind of interactions would help us ascertaining function to the unknown proteins or orphan proteins, right? Okay, Yeast-2 hybrid. So having said that, you already had Yeast-2 hybrid assay, isn't it? Did you have this thing? Okay, quickly I will tell you. So this is actually a transcription factor. Popularly, the popular transcription factor protein is Galfo protein. This is present in yeast, right? So this Galfo protein has two domains in it. One is binding domain and the other one is activation domain. So both the domains are essential for, expression of the gene that is present downstream. Okay. So what is the role of transcription factor? Can anybody quickly give me out? What is the role? It binds to a promoter and switches on the expression. So yeah, generally any transcription factor will have these two important domains in it. Okay, another key feature to understand Yeast-2 hybrid is this. Actually, in the original folded 3D structure, this binding domain and activation domain are close to each other. One exclusive feature about this Galfo domain is, even if it is separated, understood, even if the binding domain and even if the activation domain are separated, if they don't have any physical contact, then too they can still activate the transcription. It is not entirely pulling them out. When they are in close proximity, it can activate the transcription. Understood? What is the key feature? Okay. When I separate the domain, but if I maintain them in close proximity, It still can activate the transcription. Okay, so with this key feature, we are using it to identify the protein-protein interaction. So here, this is the binding domain. This is the bait protein. What is bait? What is prey? What is bait commonly? Literally, what is bait? What is bait? Like if you want to capture or catch a fish, then the worm will be the bait. What will be the prey? The fish itself. Okay, so if you have a bait for mosquito, the prey would be mosquito itself. Okay, so here the known protein is called as the bait. We know its structure function. So the genome sequence of this bait protein and the DNA binding domain is fused. I mean, we produce a recombinant DNA and hence the recombinant protein is produced. Now, if you suspect that one protein may bind to a bait protein, so the DNA sequence of that and the DNA sequence of activation domain is also fused. So we now produce a recombinant protein. Okay, so having this preparation, we can allow it. I mean, this is the common thing. Like you can see US is upstream activation sequence, a signal. You see gal for binding domain and the activation domain. In between, you have few other residues of the transcription factor. So if this attaches to US and at a particular time, the gene would get expressed. Now look at here. You have US binding domain is there, and we have a bait fused with it. So if the prey is not a partner of this bait, will it come and attach? No. If the prey is not coming and attached, the activation domain is also far away. Okay, so will the expression of this gene happen? No. For the expression of this gene to happen, BD should be in close proximity with AD, the activation domain. All right, so this is the unsuccessful experiment. Now come here. I mean, the third panel, you have upstream activation signal. The BD itself is not recognizing the UAS, okay, because of some technical errors. So this activation domain will look for binding domain. It is not there. First step itself is a failure. So this thing would not happen. I mean, the expression would not happen. Last is going to be a successful one. See, a binding domain has recognized a UAS. It is bound to it. Bait is there along with it. Now, if the bait protein is an interacting partner of prey, prey will come recognize the bait and it will touch. It will interact. So what is coming along with prey? What protein is coming along with prey? Because we have fused in a vector the activation domain. So activation domain is also coming along with prey. Now you see the setup is in such a way, this binding domain and activation domain are not directly interacting, but they are in close proximity. So is this sufficient to turn on this gene? Yes. So now this particular gene would get expressed. I mean, the protein product would be produced, and there are some assays to check if the protein product is produced or not. So if a protein product is produced, we get to know the next assumption is AD and BD were there, isn't it? They were there in the close proximity. But AD and BD are not alone, isn't it? In between AD and BD, we have fused bait and prey. So you come across, I mean, you have identified one protein to protein interaction. This is not considered as a high throughput method. At the end of one experiment, you will be able to identify one protein-protein interaction. Right? This is not considered high throughput, but it is a classical way, accurate way for identifying protein-protein interaction. Quickly to the next slide. As I told you, there are some computational methods available to identify protein-to-protein interaction methods. Phylogenetic profiles, Rosetta Stone method, a very popular method, gene neighbor identification method, co-evolution method, gene clusters method. So though you have some kind of cluttering terms, all of these rely on sequence analysis only. For all of these, the raw data will be the sequence. All right. Okay. Anyways, we are not going to look on these methods. I'm just telling that there are plenty of methods existing to identify protein-protein interaction in silico. Okay, so now databases. So having identified so many protein-protein interactions, we need to feed the data. We need to deposit the data in some databases for other members to access our data, isn't it? So interactions are collected, we put them in specific biological databases, and you can study that further. I mean, when you identify one protein-protein interaction, when you deposit in a database, anyone who is in China or anyone who is in Europe or anyone who is in America can access your protein. They can annotate it. They can cite your work. That's how the scientific community grows, isn't it? Okay, so the first ever interaction database is DIP. Okay, DIP stands for Database of Interacting Protein. This is the first ever database to hold, to store protein-protein interactions, right? Okay, so now there are several databases. So what you're going to do in your hands-on will be string database. This is also a popular database. String can give you a publishable quality protein-protein interaction data. Okay, so this string includes both physical interactions and functional kind of associations as well. Okay, so where do we, where does string gets the data from? From genomic context, genome-wide association studies, it can get interaction data. From high throughput experiments, there are several pull-down assays from which you can get protein interaction data. Co-expression is also high throughput method. And from previous publications, like whoever has identified any other protein interaction. So string also strives to get data from that publication. Yeah, for plenty of organisms it has. Okay, so this is the list of different protein interaction databases. The full form is given in the first column, the actual database name, the URL, and what does it actually contains is that. Okay, so now we have identified protein-protein interaction. We have stored them in the database. What is the next thing? We have to construct a network. How far a single protein is interacting to other protein. What is the type of interaction, whether it is a stable interaction or transient interaction, how much is it significant in the network? All this has to be assessed. So after getting the PPI data, PPI is protein-protein interaction. We build a network. After building the network, we analyze them thoroughly, right? Okay, so sources of PPI data, we have experimental data, whatever experiment would feed onto this, and intact, databases would feed, and there are meta databases and predictive softwares which can feed in the data, and we have IMEX. IMEX is International Molecular Exchange Program. So they tend to identify, collect protein-protein interaction from all of this. Experimental data, primary data, metadata, and they just have a collection from all of this. They don't identify separately. It's just a collection. So that will also feed on to the protein-protein interaction connection. So what is this? This looks so clumsy. So read this. Network visualization of human interactome. So what does this is representing? Did you at least get a vague understanding of this picture? What? All the protein-protein interactions in a human has been identified, and we have constructed a network, and this is how it will look like. Okay, it looks too clumsy, but actually that's how it's working inside us, isn't it? So there are softwares where you can zoom in, zoom out, analyze the interactions between them, which protein is strongly interacting, which protein is less interacting. All those things could be studied. Okay, a quick thing. Can you see this particular node? This particular point. You see several blue lines emerging from it. So it means that protein is interacting with many other proteins. And if you see this protein, only few lines are emerging from that, representing only few interactions this protein is involved in. That's how we should study the entire network. We have several statistical analysis method, topological metrics method to study the network in a very detailed manner. Okay. Okay. So how do we construct the network? Now we are moving on to the next step. What are the tools? We can't do it by ourselves, right? It could not be done manually. So we have softwares to construct the tools. Variety of softwares indeed. Okay. Cytoscape. This is a very user-friendly software. You can get a publishable quality metrics and images. Cytoscape, which we use it in our lab for publication purposes. Everyone could also use, like it is easy. The next one is Jiffy. Then we again have a programming. The tools that are working based on the programs. Command line called as iGraph, NetworkX, and GraphTool. These are not exclusively for protein-protein interaction, but for any kind of network construction. This is a common tool. All right. Yeah. So this is a very simple network that we could use for understanding what is protein-protein interaction. So what are these ABCD? Proteins. And what is this edges? The lines? They are the interactions. Okay. This is a simple network for understanding, for learning purposes. So commonly, a protein-protein interaction is a physical representation which shows the interaction between the proteins. And the interaction plays a key role in every process inside the cell. Okay, so of course, we need to analyze the topological features of a protein-protein interaction network so as to study whether it is a reliable network or not, or is it just a network constructed by the software. We need to assess before we understand something out of it, isn't it? Okay, so we will see what are the terms quickly. Okay, so now having a base PPI network means the mother network, okay, overall network. So that could be analyzed in 3 different ways. One is centrality analysis, like analyzing which node is essential, which protein is essential, which is the shortest path, all such things, okay? And topological clustering, like among this big cluster, you have seen no human interactome, it looks too clumsy. Could you divide them into several sub-modules? Could you divide them into sub-networks? If you divide, is it going to be meaningful or not? Isn't it? So such kind of clustering analysis could be done to the mother network. And of course, like motive search, like if the interaction is a directed network, we have 2 types of networks, directed and undirected network. So in directed networks, there are some specific list of analysis that could be done. We will see these 2, centrality and topological analysis. Okay, centrality analysis. So centrality analysis, as you can see, it's an estimation of how important a node or edge is. Like there are several measures that will tell you whether this node is important for the entire network or not. Okay, as simple as that. So this, when you perform a centrality analysis, you will be reaching or arriving at a point which will tell you which protein can be used as a drug target. After all, we are assessing the network to see if it could be used as a drug target or not, whether it is a biomarker of a disease or not, right? So overall, and when you perform a centrality analysis, you will be able to pick out, screen, you will be able to focus on at least 6 to 8 different proteins in a network, leaving the other thing as such, okay? Okay, so that's what. Centrality analysis usually aims to answer the following question. Which protein is most important and why? So you see hundreds of protein here, and you see some of the proteins are highlighted in a solid red color, okay? So after centrality analysis, you will come out with just nearly 10 proteins alone, which plays a major role in maintaining the entire network's architecture. If you perturbate, if you cut, or if you remove this one single protein, what would be the network structure? Would it be stable as before? If you remove, if you disturb, would the network stay same? No. So if you pick out, if you can pick out that specific hub protein, then it means that is going to act as your drug target, right? Okay. So centrality measures can be measured in 2-3 terms. First is the degree of network. Degree says the number of connections that it has to another node. Okay. How many connections does one single protein has? That is degree. Okay. I'm not going to go into deeper thing. Just one line explanation for you to understand the hands-on. That's all. So this is one network. How much, how many interactions does Bob has? This is Bob. How many interactions does Bob has? Let us start counting 1, 2, 3, 4, 5, 6, 7, 8, 9. So the degree of Bob is 9. That's it, as simple as that, right? Okay. So the Bob has a degree of 9. Okay, so from that, we will be able to identify the hub of a network. What is hub means? The protein that has highest number of degree is called as a hub, right? So that's what. The nodes with high degree are called hubs. So here, in this particular network, the degree has been already calculated. And which node has the highest degree? See, this particular node has 1 interaction. This particular node has 2 interactions. So 2 is given here. So as in this network, this particular node has 7 interactions in it. So the degree size is 7. So the hub would be 7th protein. Okay. So this particular protein, it maintains the entire network architecture, and hence you can relate that protein to disease as well. The disease is mainly progressed because of the expression of this protein, maybe under expression or over expression. That is a different chapter. Okay. So centrality measure, another kind of measure is closeness. So closeness is useful to measure that how fast the information would be flowing from one node to another. Okay, to understand this particular concept, look at the small architecture, network architecture. Okay, so A, B, C, D, E. Okay, so in order to understand or in order to calculate the closeness, we will have to construct this matrix. So how many nodes are there totally in this network? It's easy to understand. Don't just get scared about this formula and matrix, etc. How many nodes are there? 5 nodes. So A, B, C, D, E across the page, A, B, C, D, E listed down the page. So A to B, is there any distance? I mean, A to A, is there any distance? No, at all. So it is 0. A to B, A can be reached with B with just one edge. So 1. A to C, 1. A to D, A, B, D. 2 edges are there. So 2. A to E, 1, 2, 3. I mean 1, 2. 2 edges. Okay. So that's how we identify the path between the nodes and you fill up this matrix. All right. Okay. So, and of course, when you do the diagonal paths will have 0 value because it is to itself, right? Okay, so the total, 1 plus 1 is 2, 2 plus 2 is 4, 4 plus 2 is 6. So it is just a summation of all these things, 6, 4, 6, 7, etc. So which is the shortest path in this particular matrix? 4, okay? So when you want to pass information to the entire network, you will have to touch B node, A to B. That will give you a shortest path, okay? So the formula is actually this. N is number of nodes, where we have 5 nodes. 5 minus 1 divided by summation of Dij. All of these positions is I. All of these positions are J. Distance is what D represents. The distance of I to J is what we have filled in this matrix. So N minus 1 is 5 divided by 6. The distance is 0.67. So when you see what is the highest score that you get? For B. Okay, so that is why B is colored in red. Okay, so when you pass information across B node, it will reach the other nodes quickly. That is the motto, right? Okay, so this is the closeness measure. Okay, the next one is betweenness. Betweenness is also somewhat similar to that of closeness, but still, since the term exists, you need to know. Betweenness centrality measures how often a node occurs on all shortest path between the nodes. This seems to be same term in a very gently repeating way, but we will see this network. These nodes represent important proteins, especially like for example, C. When you bypass C, could you reach E? When you bypass C, could you reach E? Yes, A to B to D, E. When you bypass B, could you reach E? Yes, A to C, D, E. But when you bypass D, will you be able to complete the network? No. The final signal will not be arrived. Okay. So this center point or center node acts as a betweenness node for the entire network, right? This is how it should be calculated. Measuring such kind of nodes in a big network is called betweenness measure. Okay. So now we are into the last part of the network metrics. Okay. So looking for communities in a network, like we saw to a clumsy human interactome. We need to measure all of this closeness, betweenness for each and every node. That is actually a tedious process, isn't it? So now we will move on to some other way to reduce the complexity of a network. We will have to extract a small portion of a network and study that alone. Okay, so that is called as a clustering analysis. So for example, I'll tell you one thing. Velour is a big network. All right. So you can't study the entire nodes and edges of Velour. But if you think of VIT University and the quarters, we have families residing there. We have all the groceries, the vegetable shop, fruit shop, everything is there. So VIT, in and around VIT could be considered as one module, right? So that way in the entire network, you will be able to pick out a region, a sub-network, which is not dependent on the other part of the network, right? Okay. So this, when you do this clustering analysis, you will greatly reduce the complexity of the network and you can extract even functional information from that. There are several terms that is used when you extract that parts and portions of a network node. You can name it in a different way: community or cluster, module, complex, click, motive, etc. Most of the time, we will use the word module for a subnetwork. Understood? What I'm trying to tell is extracting a subnetwork from a big network. We give a name and list it here. Okay. So look at here. This is entirely one full network. And what you see delimited in orange color circle is all a module. Okay. So every module is independent. Okay, if you extract this module, it will not, it will not over-represent or oversee the next particular sub-network's function. Okay, so that's what. So a module is an exchangeable functional unit. They are self-contained and they have well-defined interfaces with other components. The characteristic feature of this module is this: its functional properties do not change. If you pick out that sub-network, place it somewhere in the network also, its functional role will not change. So it is an exchangeable unit. Understood? Yeah. Okay, so now finally, having studied all of this, sub-properties, topological metrics, all of these things, we will have to check if the entire network is of reliable, whether you can take it for further research or not. Okay, so we have to trust, before we take it for further thing, we will have to trust the network. So how can we do that? Means that we can do a MI score. Okay, MI is molecular interaction score. This method is used for assessing the reliability of protein-protein interaction data. So what are the ways we can assess if the interaction is correct or not? 3 ways. One is number of publications that says that this protein interacts with this protein. If the number of publications are more, then you can trust the interaction, right? Then, the deduction method. What method was applied to deduct? Whether is2 hybrid method or any other pull down method or Bait reverse Bait method, anything. You can rely on this particular measure. Then interaction evidence. How do we get the evidence like this is correct or not? This will all be available in the databases that we are going to work. So considering all of this, the MI score would be calculated. The greater the MI score, more trustworthy is the network. Okay, so there are other softwares which can construct protein-protein interaction. Hippie, this is also good. We use sometimes in a lab. Pina, the URL is given. Cytoscape, as I told you. It's already shown in the previous slide, one of the previous slides. Yeah, ultimate aim. Why are we much concerned? Why there is lot of budget allotted to protein-protein interaction? Actually, this is a main concern of proteomics. But when it comes to DNA function, again, in genomics, that is the main concern. A biggest challenge. Why should we focus on interaction now? You can get to know the target or biomarker of a disease. Then, what is the important concern? Why are we much concerned about protein-protein interaction? Because protein-protein interaction gives us clue about the function of a protein. Okay, because most of the protein remains as an orphan protein. We do not know why is it existing, what is its role, we don't know, but if we know it is existing, it is expressing, right? So when we perform, when that unknown protein, when that orphan protein is one among the association member, we can ascertain, we can assign some function to it. Okay, you perform this kind of role, then you belong to this family. That way we can join them in the flow, right? So that's what. So the data from PPI, the data from transcription analysis, the data from other rest of the bioinformatics analysis will give you an information. Like we will be able to assign functional annotations or even biochemical function to it and characterized proteins. Thus, orphan proteins are not left alone. We make them join in the flow, right? Yeah. Thank you. Any questions? I have tried to touch all of the important measures which we need for hands-on immediately after this session. So Cytoscape would be the better opportunity to explore the protein-protein interaction, but we will, for now, we will do it in string online. It can give you quick results than Cytoscape. Yes. Shall we end up the session, ma'am? If there is no question, should I wait? Okay. So we will all meet again in SMBG 15 to explore more about protein-protein interaction. Thank you. Yeah. Okay. So we will have to first open the string database. The website for string database is given in this hyperlink, string/db.org. Otherwise, as I told you, you can type in Google string database. Okay. So the data set for working is given in the next link. When you type 10 dot that URL, no, you have a next link called protein list. So that will be your data set. Okay, so I think for the first exercise, you don't have to use the data set. You have to just give the keyword INSR. So better I'll go. It is all self-explanatory. You can keep doing it. If you have any doubts, we have our team, Miss Nirmala, Miss Deborah, Miss Tamil Bharati, and Miss Pavitra. You can call them. We are here to help you, right? Okay. So as you open string database, by default, this window would pop up. You will have to write a protein name. So according to exercise number 1, what is the protein name that we are going to give? INSR, right? INSR, am I right? Okay, so now I am giving search. Okay, so in this first page, it will give what are the interactions, probable interactions that INSR, insulin receptor it is, has, etc. Right? So, but what is our aim? We need to see the interaction, I mean the network, right? So I am giving just continue. So when I will continue, you will see this kind of interaction map. So INSR is here, the central protein, insulin receptor. If it is insulin receptor, then what will be its first year interactor? Insulin. Okay. So you see the interactions over here. Okay. So you can see that for the same protein to protein, there are 2-3 lines, isn't it? One is in purple, one is in green. So that all tells what evidences are there for this protein to protein interaction. Either existing databases or what methods are there or such evidences. Okay. So yeah, you can have a look on this. Just read the exercise and do one by one. I'm opening the exercise 2 in the other page. Visualize the network and assess the network metrics. That is the first question. So before you assess the metrics, have a look on the network, zoom in, zoom out. I mean, that function is not available as a Java, but still you can zoom the screen by Ctrl+. Once you're done, please let me know. We will move on to the next one. This is just generating the network, viewing it. Under the network, you see different tabs called viewers, legend, settings, analysis, exports, clusters, more and less. So go to settings and change meaning of network edges. I mean, the second option given in the basic settings, click confidence. In the meaning of network edges section, change it. Like we are changing and observing how the network changes. Okay, and convert. I mean, enable this option confidence and update. Click that button update. Okay, so like this thing would be. I mean, there will be a slight difference in the network. Initially, there were 2-3 lines between two networks, but here it is just one line. Okay, so the confidence level, the existing knowledge that is not shown here. Okay, it's just one interaction. So your second thing is done. I mean, visual representation changes. So shall we do the third one? Okay. So we will see which type of evidence supports the interaction between insulin receptor and insulin receptor substrate 1. I mean, any 2 interactors you can take, not just specifically this. Okay. Okay. Can you see if you click the interaction, I'll do it again, between INSR1 and IRS1, I mean insulin receptor substrate 1, I click the edge of these two proteins. So I see what kind of evidence, what experimental methods were done to identify their interaction. See, evidence suggesting a functional link. You see here, neighborhood in the genome, I mean, none is there. Co-expression, none. Experimental evidence, I mean, experimental score is given and the score is 0.8. More than 5, more than 0.5, it is acceptable. It is more certain. Okay. If you have any readings more than 0.7, it is high confidence. 0.09, highest. Okay, we have 0.8 here. I mean, if you click this show, could you see in the last column, there is another hyperlinked text called show. If you click that again, it will tell you, I mean, what are the other data sets available out of string. Okay, deduction, confidence, etc. I mean, in BioGrid is also, the BioGrid, you see the same interaction. In Intact also, you see the same interaction. Understood? Yeah. Just explore that. Okay, now it asks which type of evidence shows highest confidence score. Which type of evidence shows highest confidence score? Which type of evidence? See, association in curated database gives the confidence score of 0.9, whereas co-mentioned in PubMed abstracts, that means previously published papers, the confidence score is 0.97. So it is this evidence which contributes so much to the confidence score, okay, which is 0.97. Like, this seems to be now simple, but this is how you should assess. It's not just we have constructed a network and this could be taken as an image, not that way. You have to assess it in all possible ways. You have to utilize the database in an extensive manner, okay? Okay. So yeah, you take time to do this. You see this confidence score for other two pairs of interactions. I mean, other two interactors. So SIM does not just take information from one existing data. It tries to pull out information from various data sources. It provides us confidence score, and then that's how it gives the data to us. Shall we go to 1.4? So to do 1.4, you will have to use the tab legend. To do 1.4, use the tab legend. Okay, so what is the question? Set of interactors can also be viewed in a tabular form, in a table. How many functional partners are shown for INSR? I mean, how many interactions are there for INSR? To see that in a table form, I mean, you have to click that legend, and at the bottom you see predicted functional partners. Can you see? So just count. Make sure that if there is anything, any link like more or less, you click that, and after that you have to count. But here there is no more option. So only these are the members, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 interactions by the protein INSR. Okay? And for each of the protein, the confidence score is given. More or less, all are greater than 0.9. Okay? What is our input? Initial input, INSR, right?
So we are seeing the interactors of INSR, or the proteins that are associated with INSR, or the proteins that are interacting with INSR. In either of the way you can interpret. So this is how we give one protein as an input. Okay. So shall we move on to second exercise? It's 1 o'clock now. So without any delay, we will move on to second exercise. So second exercise is basically to input multiple proteins. Okay. So for assessing or accessing this multiple proteins, you will have to go to the previous page. I mean, this particular page. Can you see protein list? This is the data set. So just, I mean, you will have a list of protein. Just copy this. You can even do it as Control A and C. And move on to strings homepage. Strings homepage can be reached by touching this logo. Okay, now can I trace the sequence here? I mean the list of proteins? No, previously we had one protein and that is why we pasted that one protein. But we now have a multiple data. So you will have to choose this option, multiple proteins. So after selecting, I mean copying it from there, paste it here. It will be done one after the other. All right. Okay, so now you can choose the organism as Homo sapiens. There's a pull down menu at the bottom. This one. Homo sapiens. If you do not choose also, string will somehow manage to bring out the source data. That is not mandatory, but still, this is known Homo sapiens data. So I'm clicking, making the work easier. Click search. So all the list before you're looking at the network, all the interacting partners data, experimental data would be listed. But what we are actually looking for is the network. So I'm just clicking this tab continue. So if I click this tab continue, what would be the next expected page? The network. Very good. So you will always have a large network warning when you have 500 or even 300 and more proteins. But still, just continue. String or cytoscape can handle even 10,000 proteins. When you give 10,000 proteins as your input, still cytoscape can construct the network and give you. So this is the network. It looks too clumsy, right? So some softwares where you can download and install that can assess or measure this in a very reliable way. Okay, shall I move on to the next step, or will you do it by yourself? Okay, so we have now submitted multiple protein. We have constructed the network. We will now actually, by default, the confidence score for constructing this network is 0.4. Now let us improve the confidence score. How do we do that? In 2.2, the question goes this way. Go to setting tab and modify the parameters. What is the modification that we should do is, we have to change the confidence level to 0.7. So let's do that now. Go to settings. One, two, three, four. In fourth section, you see the minimum required interaction score. So change that to 7. 7 is considered as high confidence score. 0.9 is considered as highest confidence score. So now we have increased. I mean, we are putting a stricter requirement now. So the number of proteins may decrease. It is not necessary it should decrease. It may decrease. Okay. So we should not forget to click on this update button. It's loading. Did you see the complexity of the network is slightly decreasing? Yes. So when you increase it to even 0.9, the members would decrease. Okay. Okay, so the next change that you can do is again go to settings. You will see the first option, network type. From full string network, you change to physical subnetwork, update it. You will see the corresponding changes. There will be so many proteins left uninteracted. Understood? 2.3 is the actual thing, like you can study the roles, the biological processes, molecular function of the protein, etc. I mean, even you can know the pathway in which this particular proteins are involved. So 2.3 is functional enrichment. 2.3 is functional enrichment. So how do we go do that? It is already there in analysis tab. Click that. Like after looking at the network stat, did you all look that after you change every network style, no? You can see this would be changing. The number of nodes are 537, number of edges, average node degree, clustering coefficient. I mean, clustering coefficient says how far a single node can form a cluster. Okay. So average clustering coefficient is what you see here. Okay, so below that you see the enrichment process. I mean, in what pathways, in what biological process is our set of proteins are appearing. That is called as functional enrichment. Okay. So what type of information you will see here is biological process, molecular function, in which part of the cell, I mean cellular component, the related publications, and importantly, KEGG pathways. What is KEGG? Full form for KEGG? Kyoto Encyclopedia of Genes and Genomes. So this is considered as a one-stop point for your almost all the molecular information. Okay. Okay. So yeah, the next is just a small information. How many categories of enrichment terms have you identified? How many categories of enrichment we have identified? Just count it. Tables. Look at the enriched KEGG pathways and explore the table. In how many pathways are these proteins involved? See, there is a more button at the bottom of the table. Don't forget to click that and then count. 6. 1, 2, 3, 4, 5, 6. So these proteins are overall involved in 6 pathways. Also, another take point is this: there would be some proteins or some genes which may be involved in several pathways. Is that proved here? Yes, one protein may be involved in several pathways. Okay, and in one pathway there may be plenty of proteins. That is all well known. Okay, this is how we do. Like, because of the time restriction or constraint, we are able to explore this much only. If time permits, we will do it in the future in any possible arrangements. Okay. Yeah.
If you have any doubts or questions, you can ask, or we can provide you the mail of ma'am and you can just
yes.
ask them.
Most of you are from VIT, right? Yeah. My sitting place is in Hexagon only, my office. So if you have any queries in regard to this protein-protein interaction database office, you can approach me.
Now we'll break for lunch. After lunch, at what time you have to be there?
In the POC by 2, like before 2, you need to be there because from 2,
Deborah.
guest lecture will start. Okay, so everyone, those who have registered for the lunch can go for the lunch. Outside, like there will be volunteers that can give you the tickets, or you are collection 10 years to remember. Those who are the
Green
ones
color
who are
just
registered for
distributed.
lunch. No one.
Okay.
So it's lunch break.
So we will log out on
Zoom.
Lunch is in foodies only.
How to enter the Zoom session? Sign out, right? That's
I guess it's sound. Leave.
This year's 9th National DNA Day was conducted as a hybrid event with around 300 participants joining us from 39 colleges all around the nation. Our very own event poster race was conducted as a tribute to the father of genetics, Gregor Johann Mendel. To celebrate his 200th birth anniversary, as a part of our event, we conducted guest lectures, workshops, other pre-events, and nucleo hunt to bring together the scientific and creative minds of our participants. I mention my deepest sense of appreciation and would place on record my hearty thanks to all the organizers and volunteers because of whom we made this function possible. The pillars of our event, including our HOD Asha Devi and our respected Dean Dr. R. Siva, have played an important role towards achieving our goal for DNA Day. Now I welcome upon stage Dr. Asha Devi ma'am, HOD of Biomedical Sciences, to speak a few words.
Good afternoon, everyone. I have nothing to speak, but I want to thank Professor Radha Saraswati for her wonderful job. This is the 9th DNA Day function. Let it go on, let her legacy proceeds further. And I want to thank all the student coordinators, volunteers. I've seen them daily working with some calculations, formulas, all those things. Every time they will be running here and there. Even in between classes, they used to get permission to work for the DNA Day. Congratulations to everyone, and hope you learn something from this DNA Day. This will help you for your future prospectus. And also, I want to thank the faculty members who all are involved in this program. A special thanks to everyone, as well as the SBST staff members, and a special thanks to the people over here. They were helping very well. Even they were decorating the Kutuvilakku with the flowers, all those things, which is not their job. I'm a special thanks to them. And thank you, everyone.
Thank you, ma'am, for your valuable words. I now take this opportunity to extend my sincere thanks to all the guest invites that have come from long distances for their cooperation and support. I thank them for their minute-to-minute support and guidance for encouraging at every point of time in organization of this great event. I also extend thanks to all the teachers for their cooperation during this function. Events like this cannot happen overnight. So as the wheel starts rolling weeks ago and requires planning and bird's eye for detail. Here, we are fortunate enough to be backed by a team of very dedicated and motivated team of our department who know their tasks and are very much result-oriented. Moving forward, it has been our pleasure to host all the participants of the event. The participants were very enthusiastic, and I am thankful for them coming here to attend the event for 2 consecutive days. Here, we go towards prize distribution. This is the time where we acknowledge the talent of our position holders. So for Nucleo Hunt, the team which has won the first position is Team 4. The participants were Malini, Vishnu, Jonathan, and Jyoti. For this, I would like to call upon stage Asha Devi ma'am to felicitate the team members. The it's not a team actually. So the second person who has won the Nucleo Hunt event is Harsh Pipalia. Congratulations. I would like Asha Devi ma'am to felicitate him. Thank you, ma'am. Now for the genome meme event, which was an online event, the first position holder for biomeme event is Tejas Prakash Meshram. So he'll be receiving an Amazon coupon in next 2 days. Thank you. The next position goes to Kanimoli M, who will also be receiving the Amazon coupon in next 2 days. They've not mentioned Rajiv family. Okay. Okay. Congratulations, all the winners. For the poster race, and which is also an online event, the person who has grabbed the first position is K. Kalai Selvi. And the second position holder is Manisha Prasad. Congratulations, everybody. Congratulations, all the winners, and all the best for your future achievements. Now I'd like to take this opportunity and call upon stage Dr. Radha Saraswati for the vote of thanks.
Again, good afternoon. Quite early to close this ceremony, right? So we started, and I think it was yesterday at this time we had the inauguration. So we are closing it today. Not bad, 24 hours, we have finished it. Fine, thank you. And Adeen sir, HOD ma'am, all our faculty members, professors, and there are some who were behind the scene, like Dr. Mohana Priya, Dr. Rajani Raj, who were very much eagerly doing the workshop event. I think it went very well. And then there are students behind everybody, right? And so really, I place on record, thanks to each one of you. Thank you, without which nothing could happen, right? I think we're all aware of it. That, we need 2 hands to clap, right? With one, we can't clap it. So everything is done by that. And yesterday he was talking about 5 fingers. I remember Badrinath. So I will say there are 10 here. So then only you can make a noise out of it. It is like that. So you all have joined together and you've done a big job here, right? Thank you very much. Thanks to each one of you. And like she was telling, the legacy to continue. And ma'am was also telling, thank you, ma'am. And I think we are seeing it for last, all of them were there throughout. We have been continuing. Last year we had, last year, Dr. Asha Devi ma'am was there, and before Dr. Sabina ma'am was there. So we have been having this going on. So our HODs have just helped us in taking this along. So I would see that the next year also this continues. You all will be guiding your juniors, like, your seniors guided you. I thought somebody will come here, ma'am, because they were wanting to come today. So I thought they will also be here seeing the occasion. It's okay, but and thank you participants for everything, without which nothing is possible. Without participant, that nothing can happen over here. There is God in the temple, but that God needs some followers, Otherwise, there's no use. Same thing here in a college or a university. A professor is required. Professor alone can't do the job. I can only teach. When there are no students, nothing can be done, right? So it's the same way a function is done. As a, BMG family, this is what is going on here. it has been done to the best, right? Although there will be little problem between us, that should happen. Any function at home, it happens. And then final day is such a nice day, you celebrate it, right? So today we are celebrating it a little early. So students have already arranged the tea earlier. So I thank each one of you for all your might, what you have done. Thanks. So we will pat ourselves. That's what we can do. And then today, not possible for a tea in the canteen. I don't know if madam knows, every time we go for a tea after this occasion, but I went to the food court. It is too full. So we will have it another day, right? I'm sorry for that. And that's it. And then we had a group of leaders here among yourselves. The leaders were really leading me also in the WhatsApp. So I was knowing whatever was happening was coming to me on the spot. Then followed by another group of people, right? So like I said, each one of you were a leader here in this function, right? So nothing like one couldn't do or one did a lot. All have done directly or indirectly to bring this success for this occasion, right? So very happy that everything has gone very well, and we'll close down. Before closing it, I would like to thank SBST team first, with the head, Dr. Shiva, our dean. And then everything was done with the help of VIT management. Without them, nothing is possible. So we would like to thank VIT, all of us from all of your side. We would like to thank VIT for all the help facilities given here, without which we had gone hybrid, right? Otherwise impossible for so many of us to hear from other places also. So the PRO, the finance, CTS, the estate, very much important here. And of course, our canteen people were on time giving us food, and lunch coupons were served, right? So a big thank you to each one of them. And thank you, students. Thank you, professors. So thanks. Everything has to come to an end, and it's a final here. Thank you very much, and wishing you all the best, right? Yeah. Yeah, thanks. And as a teacher, I always will tell that the speakers or with whomever you got connected here, participants, kindly have the network. Maybe today you don't want it. After 2 years, 3 years, 4 years, you might need them. That is what a function or a conference or a symposia or a workshop is made, right? Kindly maintain it and see that we can go a lot ahead, right? Wishing you all the best. Thank you. Thanks.
Thank you, ma'am, for your kind words. At last, I cannot thank everyone enough for their willingness and involvement to take on the completion of the work beyond their comfort zone. There is much wearing on the vote of thanks to express my emotion that fits the atmosphere right. I'd like to call upon stage my fellow mates Sabya Agarwal and Aanchal Pandey to conclude the event. Thank you so much, everyone.
Good afternoon, everyone. We, on behalf of whole BMG batch of 2021 to 2023, will like to present our sincerest gratitude to all our dignitaries for joining us for the celebration of 9th National DNA Day and making it a success. We thank you for being with us all this time, and it has been a great pleasure. We would like to thank our beloved Professor Dr. Radha Saraswati ma'am, our convener, who started this legacy and allowed us to be a part of it. I would also like to thank our HOD, Professor Dr. Asha Devi ma'am, for guiding and supporting us whenever we needed it.
We would also like to thank all of our faculty coordinators, Dr. P.K. Suresh sir, Ma'am E.P. Sabina, Dr. Anand Prem Rajan, Dr. J. Godwin Christopher, Dr. I. Arnold Emerson, Dr. V.G. Abhilash, Dr. V. Devi Rajeshwari, Dr. Preeti Talwar, Dr. M. Kavita, and Dr. R. Sudesh for being there throughout and guiding us. Thank you so much. And a hearty thanks to all the participants for joining us as well. Lastly, I would like to congratulate all my batchmates for successfully concluding the 9th National DNA Day and making it a part of our SPST legacy. Thank you very much. I would like everybody to stand up for the National Anthem. People. So 3 cheers to our DNA Day. Hip hip, hooray! Hip hip, hip hip. Thank you.